Hello friends, we are discussing parasexual reproduction in bacteria. We know that it occurs by three methods: transformation, conjugation and transduction. We have already discussed transformation and conjugation process in detail in previous video. You can refer my video, the link is given in the description box. In this video, we are going to discuss the third method of parasexual reproduction, that is transduction in bacteria. It was discovered by Zinder and Lederberg in 1952 in Salmonella typhimurium. In this process, DNA is transferred by virus from donor to recipient bacterial cell. Hence, what we can say that here the virus acts as a vector or vehicle for the transfer of DNA from donor to recipient, or viruses carries the DNA from donor to recipient. And the viruses that are involved are Bacteriophage virus. These infect and replicate in bacterial cells. This is because these viruses have their own genetic material but do not have the machinery. Hence, uses bacterial machinery to reproduce, that is for the replication, transcription and translation. Now let us see the steps of transduction in general, or let us see the general steps that are involved in transduction. So first of all, Bacteriophage virus infects bacteria. Here this bacteria is the donor cell. Then virus injects its genetic material and makes its multiple copies by using host machinery, that is the machinery of bacteria. And they multiply either directly by Lytic cycle, or first incorporates its genetic material into bacterial genome known as lysogenic cycle, and then this is followed by Lytic cycle when it is induced. This Lytic cycle and lysogenic cycle we will discuss in later part of this video. Then what happens? Phage particles are assembled, and these are assembled along with the bacterial genome. Either the part of the bacterial genome are packed with the virus particles randomly, that is random fragments of the bacterial genome is packed, or with special genes, that is specific genes of the bacterial genome is packed along with the viral genome in the new virus particles. These two we will discuss in detail later. Now virus has multiplied or increased its number. When virus infects another bacteria, that is the recipient bacteria, then it injects random part of the bacterial DNA, which is the donor bacterial DNA, or viral DNA plus specific part of the bacterial DNA packed in the virus particle. So these are injected, either this or this is injected. This then remains extra chromosomal or is integrated into the recipient genome. That is, recombinant is formed or recombination takes place. So their fate, whether it will remain extra chromosomal or will get integrated, depends upon the portion of the bacterial DNA, whether it is homologous or it is non-homologous, and also depends upon the nature of the bacteriophage virus, whether it is virulent or temperate. So what we discussed, that transduction occurs through two kinds of life cycle of bacteriophages, Lytic cycle and lysogenic cycle. That means bacteriophages has two types of life cycle, Lytic and lysogenic cycle, which results in the transduction. Let us see this Lytic and lysogenic cycle in brief. We will discuss it in detail in the bacteriophage video. So Lytic cycle occurs in the virulent phase. Bacteriophage virus infects the bacteria. It injects its genetic material, then it takes over the bacterial machinery for reproduction. Now what happens? New viral particles are formed. These viral particles are then released by the lysis or rupture of bacterial cell. This is virulent bacteriophage virus. It infects bacteria and injects its genetic material. Now this takes over the machinery of bacterial cell for reproduction. So it starts multiplying, as a result of which new phage particles are formed. Then these phage particles are released by the rupture of the bacterial cell. Second is lysogenic cycle. It occurs in temperate phase. Bacteriophage infects bacteria, then phage DNA integrates into bacterial DNA. This phage DNA remains intact and replicates along with bacterial genome over many generations. When induced, this viral genome is excised and then it enters into lytic cycle. Then it starts reproduction using the host machinery or the bacterial machinery. New viral particles are formed and these are then released by lysis or rupture of bacterial cell. So this is the temperate bacteriophage. It infects the bacteria and injects its genetic material. Since it is temperate, so this viral genome integrates with the bacterial genome. For several generations, it remains intact and replicate along with the bacterial genome. Sometimes it is induced in the presence of UV radiation or certain chemicals. Then this phage genome is excised and it enters the lytic cycle. That means it takes the host machinery and starts reproducing. Then new viral particles are formed and these are released by the rupture or lysis of bacterial cell. Now let us see types of transduction. There are two types of transduction: generalized transduction and specialized transduction. Let us see them one by one. First is generalized transduction. It is shown by virulent phase in lytic cycle. So in this virulent phase infects bacteria. This bacteria, it is the donor cell and injects its genetic material. Then it takes the host machinery for reproduction and forms the new virus particles. In this process, what happens? Bacterial genome, it is divided into fragments. And when the packaging of virus particles takes place, then few bacterial genome fragments are also packed by mistake. And these fragments are random. So what we say that bacterial genome packing is randomly. When new viral particles that are formed are released, in that case, if the phage or the virus that carries the bacterial DNA fragment, that is DNA from the donor, comes and infects another bacterium, which we call recipient, then it injects its genetic material in the recipient bacterial cell. After that, this injected genetic material either integrates with the recipient bacterial genome and results in the formation of recombination, or it remains extra chromosomal in the cell like plasmid. So this is the virulent bacteriophage. It comes and infects this bacterial cell. This is the donor bacterial cell and injects its genetic material. Then it takes up the host machinery and starts reproduction. Bacterial genome is also divided into fragments. When the packing of virus particle takes place, then some of the bacterial DNA fragment also get packed in the virus particle. Once the virus particles are formed, these are released from the donor cell. When this virus carrying the fragment of bacterial genome comes and infects another bacterial cell, this is the recipient bacterial cell. It injects its genetic material inside the recipient bacterial cell. It either remains extra chromosomal as plasmid or it integrates with the recipient bacterial genome. If this region is homologous to any region of this recipient bacterial genome, then we say recombination has occurred and it results in the formation of recombinant. Second is specialized transduction. It occurs by temperate phase in lysogenic cycle. So temperate phase or virus infects bacteria. Here we call it donor bacteria. Then it injects its genetic material in the donor bacterial cell. This phase genome gets integrated with the bacterial genome, and this integration is site-specific recombination. Now the phase genome and bacterial genome is together known as prophase. This is because the virus is temperate phase, hence it shows lysogenic cycle. Then the phase genome remain intact for many generations. When it is induced in the presence of UV radiation or chemicals, then it enters lytic cycle. How it enters lytic cycle? Phase genome get excised along with specific genes of bacterial DNA, because the recombination was site-specific, or the integration was due to the site-specific recombination. It then uses the host machinery to reproduce. Hence, new viral particles are formed, and these new viral particles have phase genome plus specific genes of donor bacteria. Now these viral particles are released when any virus infects another bacteria known as the recipient bacteria. Then it transfers its genome to bacterial cells, which include both the phase genome plus the specific genes of the donor bacterial genome. So now here this genetic material is transferred from the donor bacteria to the recipient bacteria with the help of virus. And here only the specific genes of donor bacterial genome is transferred. So it is known as specialized transduction. Once it is released inside the recipient bacterial cell, then it results in recombination, or recombinant is formed. Now let us understand this with the help of diagram. So this is the temperate bacteriophage. It infects the donor bacterial cell and injects its genetic material. Since it shows lysogenic cycle and hence the phase genome gets integrated with the donor bacterial genome, it shows site-specific recombination and it remains intact for several generations. Due to some cause, if it is induced by UV radiation or the presence of any chemical, then it gets excised, and during the excision of the phase genome, some specific genes of this donor bacterial genome is also excised. This is because this phase genome is integrated into the bacterial genome by site-specific recombination. Now it enters in the lytic cycle. It uses the host machinery and starts reproducing. New viral particles are formed, and all the viral particles has phase genome plus specific genes of the donor bacterial genome. These are released when this virus comes and infects another bacterial cell, which we call recipient bacteria. It injects its genetic material into the recipient bacteria. Then this genetic material gets integrated with the recipient bacterial genome. Either the entire genetic material containing the phase genome and the donor bacterial genome gets integrated, or only the donor bacterial genome gets integrated with the recipient bacterial genome. So in either case, recombinant is formed, and this transfer of the specific genes of the donor bacterial genome to the recipient bacteria is carried by virus, and hence it is known as specialized transduction. So this is all for today's video. If you like this video, please hit the like button, share it and subscribe my channel. Thank you.